Gurunathan for providing me an opportunity to conduct this webinar. Foremost, I welcome and thank my principal ma'am, Dr. R.R. Jayanti, Principal, Government Arts College for Women, Krishnagiri, who has been a keen instigator for my workmanship, and encouragement for conducting this seminar. He is being the pillar and a keen operator for the faculty improvement, where I thank her for this in this opportunity. Good morning, Principal Ma'am, my faculty participants and student participants and scholars participants for this webinar series. Of all, I welcome Dr. T. Vinod Kumar, who has been the keen person of motivation to conduct this webinar by myself. He has been the supporter for this. It is my immense pleasure and more excited to welcome everyone. On behalf of my department staff members, Dr. Srinivasan, Dr. Murli, Dr. Kanaglakshmi, Dr. Tamarai, Dr. Sridevi, Deepa, Tamarai Chelvi, Shanmu Priya, Shobhana, of all the team, the committee members, it is my part of my pleasure and gratitude to welcome everyone. I also welcome the participants who have been so keen interest and showing the response the day when I opened the link for registration. Almost 100 participants linked in the same day for registered their request, showing their thirst and interest on the subject topic. Even in a difficult pandemic environment, they are showing such a keen academic response. I really appreciate and welcome everyone. This Google platform shall meet you with a renowned speaker, an excellent speaker, associate professor, who is a sincere, hardworking, always updating their knowledge, young and dynamic scientific professor, and a very good administrator, Dr. T. Vinod Kumar, associate professor, School of Biological Science, CMS College and Commerce, Coimbatore. I would like to present one small keynote of Swami Vivekananda and then lead the platform to Dr. T. Vinod Kumar. Swami Vivekananda has told, நாம் இன்று எப்படி இருப்பதற்கு நாமே பொறுப்பு. இனி எப்படி இருக்க வேண்டும் என்று விரும்புகிறோமோ அப்படி நம்மை செய்து கொள்வதுதான் ஆற்றலும் நம்மிடமே உள்ளது என்று சுவாமி சொல்லியிருக்கிறார். இது ஏன் இதை படித்தேன் அப்படின்னு சொன்னா, இது டாக்டர் டி வினோத்குமாருக்கு பொருத்தமானது. அவர் எங்க இருந்தாலும் எந்த என்விரான்மெண்ட்ல இருந்தாலும் தன்னை வளர்த்திக்கிறதும் இல்லாம தன்னோட என்விரான்மெண்ட்டை அப்கிரேட் பண்ணி டெவலப் பண்ணி அதுல ஒரு ஸ்டாரா இருக்கிற எண்ணம் கொண்டவர். அவருக்காக இந்த நோட். தென் அடுத்தது நம்ம எல்லாத்துக்குமே பொருத்தமான பாரதியாருடைய நோட். மனதில் உறுதி வேண்டும், மனதில் உறுதி வேண்டும், வாக்கினிலே இனிமை வேண்டும், நினைவு நல்லது வேண்டும், நெருங்கின பொருள் கைப்பட வேண்டும், கனவு மெய்ப்பட வேண்டும், கைவசம் அது விரைவில் வேண்டும், தனமும் இன்பமும் வேண்டும், தரணியில் பெருமை வேண்டும், கண் திறந்திட வேண்டும், காரியத்தில் உறுதி வேண்டும், பெண் விடுதலை வேண்டும், பெரிய கடவுள் காக்க வேண்டும், மண் பயனுற வேண்டும், வான்மிகு தென்பட வேண்டும், உண்மை நின்றிட வேண்டும். இது எல்லாமே நம்ம யங்ஸ்டர்ஸ்க்கு, த பட்டிங் சயின்டிஸ்ட்க்கு, டீச்சர்ஸ்க்கு ரொம்ப முக்கியமானது. இது பாரதியாரோட கோட்டு. இது வித் திஸ் கோட் ஐ லைக் டு இன்ட்ரோடியூஸ் Dr. T. Vinod Kumar, the dynamic scientific professor who has been completed his qualification BSc Biochemistry, MSc Biochemistry, MPhil Biotechnology and PhD Biochemistry. In addition, he has also qualified PGDCA in 2001 and 2. He is right now working as an associate professor in CMS College, Science and Commerce, Coimbatore. but he has got experience as associate professor, faculty of medicine, medical technology, Misurata University, Libya. He has been abroad for 5 years. He has also worked as a senior lecturer earlier, and he has been my colleague in 1995 to 1998 in Sri Ner Mahavidyalaya College of Arts and Science, Coimbatore. He has shouldered lot of awards, publications. He's got a CV of 18 pages where time is limit. I wanted to read out the important points which will attract all the participants. He is now being a director as well as head of the Department of School of Biological Sciences at CMS College. His research experience is drug discovery, screening, extraction, isolation, purification, and characterization of naturally occurring secondary metabolites as enzyme inhibitors as a future drug candidates for diabetes mellitus, Alzheimer's disease, atherosclerosis, and cancer. He is also bioinformatics, molecular modeling and docking strategies, prediction of binding site, interaction with functional groups, QSR studies, molecular modeling and comparison of silico binding energy with in vitro analysis. He has got also the experience in bioprocesses technology, screening isolation in bioprocesses, and feed batch and continuous fermentation of fermenters, etc. Upstream and downstream processing also is well known. Research projects, he has obtained MSc Biochemistry research project entitled A Study of Alpha Amylase Inhibitors of CO4, was guided by Kai Manavan, Professor of Head and Department of Biochemistry, Center of Molecular Biology, Tamil Nadu, Agriculture University in 1994. Research outcome, he has delivered thermostable and antimicrobial alpha amylase inhibitor from Cetaria italica Len. MPhil Biotechnology, he has completed dissertation on study on cholinesterase inhibitors from Bacopa in Mominaria Lin and Clitoria terminatia under the guidance of Dr. R. Bhupati. He is a retired professor from Bharatiya University. And he completed PhD biochemistry on novel cholinesterase inhibitors from Indian medicinal plants with relevance to Alzheimer's disease. He is responsible for publications, more than 25. More than 25 publications are impact factor with index more than 40 something. I'm not sure about that, but almost all the 20 publications are on ISSN publications with very good impact factor. And he has published 2 books. He has published 2 books on clinical biochemistry for clinical biochemistry tools and diagnosis, and published a molecular diagnosis book for biotechnology. Excellent, sir. And he has got patenting on the characterization of the beta amylase compound cholinesterase antagonist in the year 2010. He has also significantly highlighted about his research. He has published the 16S or DNA gene sequence of novel antibiotic producing streptomyces genome in the year 2010, and also published the same gene sequence of novel cholinesterase inhibitor producing streptomyces in the very next year. He is the author for the gene, NCBA gene, accession number F4824089. So far, he is an supervisor for MSc projects of 55, MPhil guided around 7 candidates, PhD candidates around 6, and students completed PhD, 3 of them under his supervision. With a very eminent, very scientific and academic notes of curriculum vitae, I welcome once again Dr. T. Vinod Kumar for immediately accepting my request and for the presentation in the today's webinar series. I welcome, sir. Kindly take over the charge.
Thank you, madam.
I request all the participants to pin your slide during the presentation. Note down all your points of questions. At the end of the session, you are given 30 minutes time for interaction. That time you can open the speaker and please share your queries. Thank you, participants. Thank you, principal ma'am. Thank you, my staff members. Welcome, sir, for the presentation.
Thank you, ma'am. You should not take this much time, okay, for the introduction. I'm a very simple man, okay, I have to go, okay, okay. And because you are the inspiration, okay, for me. And so with your friendship and with your guidance, we excelled so much, including you and Sudha ma'am, okay, all the seniors of PhD, Arts and Science College, okay, from my department, and you guided a lot, okay, to this stage, and still we are growing each and every day. Thank you, madam. Thank you for your introduction, and thank you, Principal ma'am, Jayanti ma'am, okay, for your presence here, and thank you all the faculty members of PG and Research Department of Government Arts College for Women, Krishnagiri, and okay, all the fellow colleagues of biochemistry fraternity, and also my dear students, okay, for this wonderful webinar session. And this is not going to take okay much of your time. I know, okay, you're all students, okay, maybe you can withstand, okay, maybe 30 to 40 minutes to clear observation about, okay, my talk. Okay, I'm going to limit, okay, my talk with maximum 40 minutes, okay. Thank you for your presence, everyone. And this small quote, okay, again, by Swami Vivekananda said by madam. So we have to do our best, and automatically, okay, God will give the best to us. Okay, that is the duty, the karma. So karma, if you are doing, automatically the God will give everything to us. So each and every day is assigned to us. So based on that, we are doing our duty. So this is my great honor to be here in this forum today. So that is a decision of God, and I'm giving the best to you, the knowledge. Thank you, Andal. And it's my duty to start the session, and I'm going to share the screen. So just to pin the screen. I think you can see this window. Can you able to see the window? Yeah, if you able to see the window, just pin it. So there is a pin button in the center screen. Just you can pin it. So if you're not able to see the window, maybe you can check. So
yes, sir. Yes, sir.
Okay, right. So my topic for today's webinar of so Government Arts College for Women is Peptide Microarray and COVID-19 diagnosis. It is not a new topic. So this was the concept was discovered, okay, more than 10 years back. And so this one is developing each and every day, okay, this topic. And this concept is unique nowadays because now we are in the corona condition, that is COVID-19. And we need an alternative tool for the diagnosis of COVID-19 because at present we are using qPCR, and this qPCR is time consuming, Sometimes, okay, if it is not done properly, so that may give false positive results and also false negative results. That is that also possible. And this is an alternative. So this technique is developed by a German company, the Pepperprint, and they are doing a lot. So only because of this technique, okay, they did so much of achievement in screening the people of Germany, and okay, now they succeeded to control the COVID-19 condition. So we hope in the near future, okay, we can also do this one. And with the small introduction about a microarray, because some of the students from the PG level also, just it's my duty to give the introduction to them. And for the faculty members, that may be very basic. So please bear with me. And so this is the molecular diagnostics. Coming to the molecular diagnostics field, okay, this is one of the new era, okay, in the diagnostics. The molecular diagnostics, we are going to use molecules like DNA, RNA, and proteins, and some of the small molecules also. So my focus today is only proteomics part, because I'm not going to cover okay much of genomics. Okay, there is another ocean okay in molecular diagnostics. You can see this slide. Okay, here the conventional method. Usually, a person suffered by cancer. So initially, maybe for 10 years back and all. So we have to give the sample, the suspected person sample, that is called as biopsy sample, okay, to the hospital, and they will analyze the person. The responsible for the analysis is called as a histopathology person, and the pathologist, he will analyze the sample by taking the tissue sectioning with the proper staining and all. He will identify based on the abnormal cell, okay, that's called as cancerous cells, okay, based on its morphology, because normal cells are having the normal morphology, and if it is a abnormal cell, okay, they may have the different characteristics. So coming to the molecular diagnostic method, nowadays, okay, we are not dealing with the conventional method. So maybe for the preliminary screening, okay, we can identify, okay, using this conventional method. And nowadays we are using the molecular diagnostics, either proteomics or genomics. So this proteomics is very important, because we are going to use the protein tools, the proteins as analytes. So we can identify some of the marker proteins, okay, in the patients. So I'm not focusing about, okay, here COVID-19, okay, this is generally a disease and a normal person. So in the normal person, okay, some of the proteins will not be expressed. At the same time, the abnormal person may be in cancer, or otherwise infected by corona, or otherwise malaria, or whatever may be, okay, any abnormality. So this may lead to express some of the proteins of unknown origin, okay, that is called as, okay, these proteins are expressed only in the disease conditions. So we can identify the proteins by 2D page, and based on that we can identify, and based on the further research we can do something, okay, beyond that, beyond the 2D page analysis. So that is called as microarray. Initially this microarray is very specific to DNA, because coating the DNA on the plate is very easy when compared to the proteins, because you already know that, okay, the studying the proteins are very difficult when compared to the molecular biology, like dealing with the DNA and RNA, because they are all more stable, okay, than compared to proteins. Because if you are heating it, okay, the DNA may be separated into 2 single strands, that's all, but the denaturation will not happen. And here in the proteins, if you are keeping the protein in the abnormal temperature, like maybe the hot environment, so this protein may be denatured, it loses its property. So the dealing with protein is a big challenge, okay, that is why a lot of demand, okay, for this field. Those who are excelling, okay, in this proteomics field, they can get a good scope. So this is the future of biochemistry. And coming to the my topic, okay, this microarray. So here I'm going to tell about first of all the DNA microarray, and after that we can go to peptide, because you need some knowledge, okay, to gain. So I can start with the DNA microarray, and this is the biochip. You can see a person holding the black color chip. So the chip having the central part, okay, is a small the glass or quartz plate. So this is called as biochip containing a microarray. So this is the overall the view of, okay, this one. So based on the definition, the biochip is nothing but it is a miniature test sites, okay, arranged in a solid substrate, okay, maybe in a plastic container or a plastic card or something. And this facilitates, okay, many assays to be performed at the same time. So for example, if you are giving the blood sample, okay, for serum albumin or otherwise bilirubin So any one of the analyte, okay, maybe they will do the assay for only one time. So by using this chip, you can do the maximum, the limitation is 2,40,000. So that means you can do 2,40,000 tests, so within a single time. Okay, that is advantage of biochip. And I said, okay, earlier, the biochip is already having microarray. This microarray is nothing but it is ordered array of nucleic acids, proteins, small molecules, including now the small molecules are applied in this field. And this will facilitate the parallel analysis of the complex biochemical samples. So this is an introduction to biochip and microarray. Next one is the development. So who are all responsible for the development of biochips and microarray? Don't think only the biological scientist, okay, maybe the microbiology person or biochemistry or biotechnology person responsible. So no, it is a cumulative effort of okay, all the persons involved. So like electronics and nanotechnology, bioinformatics and pharmaceutics. So all the four disciplines are combined together with the background knowledge or the basic knowledge of the biochemistry, molecular biology, genomics, glycomics, and proteomics. So this is the integral part, okay, combining all the fields like electronics, the bioelectronics to scan the chip, and the nanotechnology to implant, okay, the molecule, okay, on the slide, and bioinformatics, we need the data interpretation, and the pharmaceutics, the application of the microarray for the diagnosis part. So next one is the evolution of microarray technology. So I said, okay, maybe only 10 years back. So the only 10 years back, it evolved into the molecular diagnostic area. But actually, in the year 1990, Dr. Pat Brown, and he's a scientist of a Stanford University professor, and he developed a technology with a small glass slide-based microarray. So based on this, he developed a patent, and the patent is distributed to all the companies. The one company started the microarray technology as a diagnostic tool by using the photolithography to implant, okay, that DNA samples on the plate. The company name is Affymetrix. So they are the leaders, they are the pioneers, okay, in this field, and still, okay, they are dominating it. And this is just of the chronological arrangement of the achievements of maybe the discoveries of microarray technology. And 2004, the new terminology is introduced, okay, in this field, okay, that is called as the clinical bioinformatics. We all know that, okay, we already studied all the biochemistry students, the clinical biochemistry may be studied, the microbiology students, okay, they may be studied, okay, the clinical microbiology. Okay, but so we have another area, clinical bioinformatics. So this area focuses, okay, mostly on the informatics, that is the database and data and collection and interpretation related to the disease, okay, that is called as clinical bioinformatics. And the major vendors, okay, at present, okay, we have so many vendors, okay, for DNA microarray, till now, okay, the same leader, the Affymetrix, he is belonging to Santa Clara of USA. And Agilent Technologies, they're also pioneering in this field, Applied Biosystems and Combimatrix, GE Healthcare. So all the five dominating, okay, in this DNA microarray field. And coming to the types. So now we had an idea, okay, what is a biochip and what is a microarray? I hope, okay, you understand the concept, okay? This may be new to you if you're a PG student. And maybe now you can have an idea what is a biochip and what is a microarray. And this microarray is the common terminology, and we have the different types of microarrays. The first one is DNA microarray, second one is protein or peptide microarray, glycan microarray, and chemical or small molecule microarray. So DNA microarray, in the microarray is a small chip, okay, the glass or otherwise quartz chip. In the quartz chip, okay, we can label or otherwise fix the DNA molecule, okay, that is called as a DNA microarray. If you are using a protein or otherwise a peptide, that is called as peptide microarray, and a glycan chain, that is a carbohydrate chain, if you are attaching it, okay, that is a carbohydrate microarray. And the last one is the chemical or small molecule microarray. A lot of research is needed, okay, in this area. Only the biochemist, okay, can solve this lot of glitches, okay, in this field, the fourth field. If it is successful, okay, maybe we can have a clinical biochemistry lab. So within a small room, maybe the size of, okay, 10 by 10, 10 feet into 10 feet. So we need only, okay, 2 or 3 microarray machines. The problem is, okay, each and every microarray machine may be 2 to 3 crores. So that is a main drawback. But if you have, okay, that one, you can do maximum possible, that is a 2,60,000 tests in a single run. For example, if you want to give a blood sample, okay, to the clinical biochemistry lab, and you can request maybe that particular one, the requirement, okay, the glycan, maybe the blood glucose level, fasting or otherwise postprandial. So based on that, so this one is each and every test, okay, you have to request it. But here in the chemical or small molecule microarray, so you can have a lot. Okay, that means you can conduct all the biochemical parameters in only one test, okay, within 2 to 3 hours of time. So we can get a complete profile of a man, whether he is normal or abnormal, and whether all the biochemical parameters are normal, whether he is having some abnormality in the liver function or otherwise kidney function or otherwise maybe the hormonal imbalance or any other metabolism related to maybe the vitamins or minerals, whatever may be. And this is the last one. Still, it is growing day by day, and the contribution from the biochemistry field is required, okay, for this. So till the research work is going on, okay, in the last one, and all the three, the remaining, the first one, second one, third one is completely grown. And the DNA microarray is well developed, and protein and peptide microarray, they're developing a lot, okay, nowadays, and the glycan microarray is also doing a lot. So we can go with one by one, and so first one is DNA microarray. Here the sample is used as a DNA microarray is
not visual.
yeah, the slide is visible to everyone. If it is not visible, just you can pin this the presentation screen from the side. There are two Vinod Kumar will be there. Just you can pin it. I hope everyone is getting the slide. Maybe somebody started presenting it. Okay, that's why the format is
visible, sir. Visible,
yes.
sir. Vinod sir, it's visible.
Okay. Okay, and the sample is a DNA sample, okay, in DNA microarray, and so usually here we are not using the double stranded one, only the single stranded DNA, because you already studied about the hybridization. Hybridization is a process, one strand, if it is complementary to the another, okay, they may hybridize. So this hybridization is possible only with the single stranded DNA. So we need the single stranded DNA. In the case of single stranded DNA, we can create a DNA, okay, that is a complementary DNA can be produced from mRNA, so for the cancer diagnosis, and this can be easily produced, and this cDNA microarray is very common in cancer diagnosis. Just have a look, okay, in the PPT screen, DNA microarray, the first slide is all the single stranded DNA molecules are plated, okay, on the microarray plate, and the complementary nucleic acids with maybe the fluorescent tag, okay, can be placed, and that may having the interaction with the single stranded DNA that form hybridization. If it is hybridized, okay, automatically that will give the signal. So the fluorescence. So the fluorescence can be identified by a scanner, simple scanner. So you can identify, okay, what are the spots giving the color, if it is green color or red color or yellow color. So we are going to see the what is the color pattern, okay, in the next. Second part is the protein or peptide microarray. The sample used here protein. Usually the proteins are very large one, and handling the protein is very difficult, and the proteins are easily prone to or otherwise maybe denatured, okay, by heat or any other chemicals, okay, including the salt concentration also. So those are studied biochemistry. You can understand, okay, this complexity of handling the proteins. So for that, okay, we have the alternative, okay, that is peptides. A small fragment of proteins can be used, and usually, okay, all these peptides are not maybe derived from protein. So this is synthesized, okay, by the technology that is called as that is a peptide synthesizer. So there are machines are there, and these machines, okay, will produce a lot of peptides, and the peptides can be implanted on
Sorry to interrupt.
micro.
PPT
Yes,
is
madam.
not shown.
PPT is not found.
Yes.
It's clear, madam. Maybe let them choose it from the right side. Somebody presented, madam. Okay, that is why the problem happened.
Kindly, participants do not present now. Tamarai Chelvan, some candidate participant has interrupted. After that, PPT is vanished.
PPT collapsed. Students, go to the section. Maybe the top 137 is there. Okay, just go to the show everyone, and after that, go to Vinod Kumar. So there are two T Vinod Kumar will be there, and one is the presentation screen, and another one is the slide. Okay, and there is a pin symbol will be there. Just you can pin it, and if you are pinning it, somebody is coming accidentally. Okay, by presenting the screen, so this will not be disturbed. So please do it. Yeah, I have pinned. Thank you for your response.
Till now, PPT is not shown, sir.
For you, madam?
Yes, sir.
Maybe you can also follow the same steps, madam. Just go to the top. Show everyone will be there. 136 or 137 number is there, madam.
Okay.
Just you can press it.
Okay.
Then go to alphabetical order, Dr. DR. And Dr. T. Vinod Kumar, bracket biological sciences. There are two T. Vinod Kumar will be there. Just you can scroll it, madam, to the D. It is alphabetical order. Can you able to see it?
Okay, continue, sir.
You got it, madam?
I will try. I will make it. Continue.
So simple, madam. Just you can go to people and go to T. Vinod Kumar. There are two T. Vinod Kumar will be there. One is my presentation screen and another one is my video. Just
Okay.
pin it. Okay, the T. Vinod Kumar. You got it?
Yeah.
Just pin it, madam. The box like that you can pin it. So this is a problem in Google Meet. If somebody is coming and presenting, okay, my presentation will be collapsed. Okay. I hope everybody is seeing my window, right?
Yeah.
I hope. So I'll continue with the peptide microarray. Just we have the difficulty of using the proteins as a whole, okay, in the microarray chip, because proteins are very sensitive to the temperature and maybe very easily prone to the proteolytic enzymes. If it is the plate is contaminated with enzymes, okay, they may be degrading the protein. At the same time, okay, the water, what you're going to use it, okay, if it is not buffer, automatically, okay, this one will denature the protein. So a lot of complications with the protein handling of proteins. That is why, okay, now we are going to the peptide part. So peptides are a small fragment of proteins with very limited numbers, okay, maximum 10 amino acid sequence, okay, that can be tagged on the microarray plate. So that may be used for the further investigation. So we are going to come to the later part, okay, for advanced studies. And the subtypes are called as enzyme microarray. We can tag enzyme also, antibody microarray, protein microarray. So this one, the enzyme microarray and the antibody microarray, the protein microarray, not very famous. The big problem is the handling of the proteins, because proteins are very big, okay, in nature. So now we are going to use only the peptides, okay, in this segment. And third part is in the classification. The third classification is the glycan or carbohydrate microarray. The carbohydrates can be tagged on the microarray plates, and if you have the specific antibody with the conjugated with enzyme or otherwise conjugated with some fluorescent particles, so you can see, or otherwise you can identify the spot whether your glycan is present on the microarray or not. So this is very important, okay, maybe if you are dealing with structural glycoproteins, because mostly, okay, the proteins are made up of not 100% proteins, maximum, okay, maybe 24 to 25% or 26% of the glycan component may be there, okay, for the fully functional activity of the protein. And the carbohydrate can be used in the form of structural glycoproteins, example, receptor protein or the secretory protein, and the glycoprotein enzymes, mostly all the enzymes with the functional properties like binding or interaction. So all these, okay, having the glycoprotein component. The microbial cell membrane glycoprotein, okay, that is also very important. So this is also another area, glycomics. Glycomics means study and characterization of, okay, the carbohydrate moieties. Yeah, The next one is just now I said, okay, the chemical and the small molecule microarray. This small molecule is nothing but, for example, analytes. For example, if you want to analyze maybe a particular analyte, for example, bilirubin or biliverdin or otherwise creatinine, okay, or anything, okay, related to the biochemical focus. So you can create a chip, and within that there may be a microarray, the microarray having all the components. So still it is growing. This field is not very easy because we can handle the peptides, DNA, okay, like this maybe very easily. Tagging is not a problem. But the chemical molecules having each and every maybe the property, the tagging or attaching the chemical molecule on the microarray plate is the very difficult task. So attaching all the different analytes of biochemistry on a single chip, it is very difficult. Now the technology is researched by so many the pharmaceutical companies, and maybe very soon, within 10 years or 20 years, okay, we'll get a small chip, and maybe within a 10 by 10 room, we can do all the analysis, all the biochemical analysis. There is no need for going to the clinical biochemistry lab. Yeah. So these are the stages of DNA microarray. I'm going to explain about DNA microarray now. Then later stages we can go to the peptide microarray. This is very simple. We have to use a DNA microarray. So usually this DNA microarray cannot be developed by you in the lab. So this need so many robotics equipments to tag or otherwise to attach the DNA, single stranded DNA on a microchip. So the manually it is not possible, and you need so much of technology. So that is why I told, okay, the nanotechnology is required. And extraction and purification of mRNA from the clinical sample. For example, if you want to do, I'm going to say an example, okay, the cancer diagnosis. So you are going to analyze, okay, this sample of the person is whether he is suffering from cancer or not. So that is normal, and also the clinical sample from the cancer patient. So we can extract the mRNA. From that mRNA, so you know the process of copying the mRNA into DNA. So that is called the cDNA synthesis. cDNA is nothing but complementary DNA synthesis. So this complementary DNA synthesis, you can take two samples. One is normal sample and another one is the cancer sample. For the normal samples, you can use the cDNA machinery, okay, that is the reverse transcriptase enzyme, okay, that will produce cDNA. At the same time, all the nucleotides, okay, what you are going to use can be attached with the cyanine 3, okay, the COA3 molecule. So this COA3 is a molecule that will fluoresce in green color. So the green means normal. So that is why for identification, we are going to use COA3, okay, to the machinery or the cocktail of cDNA synthesis of normal samples. And this is readily available, okay, no need to make, okay, this one. And for in your case, okay, the patient gave the sample, and from the patient you are going to get mRNA, and from the mRNA you are going to synthesize cDNA. At the time of synthesizing the cDNA, the complementary DNA, you can use cyanine 5, okay, that is the COI5 molecule. So this COI5 will fluoresce, okay, in the presence of UV light, okay, this will fluoresce into a red color fluorescence. So this red color fluorescence is an indication of abnormality. Okay, that means we need some green and red, including for the signals. Okay, we are using the same colors, right? Okay, same thing. So this RT-PCR is very useful. Okay, for this maybe for this experiment, and hybridization of cDNA probes into DNA microarray. Just you can add the cDNA, both normal and also abnormal one, that is green color and red color, the mixer one, okay, to the microarray, and detect the hybridization signal, okay, after hybridizing. DNA microarray data analysis can be performed. For this, okay, I'm going to show you a small video. So this video will give you whatever, maybe I can tell you nearly 1 hour lecture, okay, to you. You cannot understand the concept unless, okay, you see the methodology there. So that is why, and I'm going to see the demo, show you the demo, the DNA microanalysis. So this is a video. So this video, I'm going to give the audio also.
DNA microarrays, sometimes called DNA chips, reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously.
Yeah, this is a very simple demonstration, the animation about the DNA microarray. I hope okay everybody understood the concept of DNA microarray. So in the previous demo, they used yeast cell placed in aerobic condition and anaerobic condition. We can identify what are the genes are expressed in aerobic condition and what are the genes expressed in anaerobic condition. And the yellow color is the genes expressed in both the conditions. That means both anaerobic and aerobic. So this is a very simple one, and we can use the same technology for cancer diagnosis. And this is the anatomy of the DNA microarray chip. Just you can see here, the 1.28 centimeters into 1.28 centimeters, this is the actual size of the gene chip. And within that, approximately 5 lakh cells can be possible. That means you can attach 5 lakh different types of DNA strands. Okay, and each and every spot may be represented in the small square, and they may be expanded okay to this level. Okay, you can see a small single stranded DNA is placed, and the maximum size of the single stranded DNA allowed okay in the microarray chip is approximately 25 base pairs. So we can have up to 25 base pairs okay in a single stranded DNA. So this is the anatomy of DNA microarray. So next one is you have to understand okay what is hybridization. Hybridization means the complementary DNA binding. Okay, this is a single stranded DNA, and if you are tagging with a red color cyanine molecule, okay, this CY5, and this DNA, the complementary DNA is complementary to the already the tagged one, means okay this will form a hybridized form. Okay, that is called as hybridization, and this will form double stranded DNA. Okay, this is a technology, the scanner, okay, the scanner of this microarray having both wavelength. So one is CY3, okay, that is 532 nanometer. As a biochemistry students, and you're all very known to the colorimetry and all. So you know very well about okay Beer and Lambert's law. So no need to explain what is the role of nanometer and all. Okay, the 532 nanometer and this is 635 nanometer. So both will detect the two colors, green color and red color. And if maybe a spot is having okay both the expression, both CY3, that is the green color, and red color, means so that will give the blue color, sorry, yellow color. So this yellow color represents the combination of both. Okay, and you can see this is the final result. And finally, okay, you will get a green color one. So this greenness may be healthy, and a red color means okay that is the diseased one, and the yellow color is so these genes are expressed in both the conditions, okay, both normal and abnormal condition. And this one is so to check okay whether your strand is working or not. Okay, that means some control parameters so can be used okay that will not give any fluorescence. So I hope okay you can understand okay this concept of microarray. And I'm going to the instrumentation part. So these are all the instruments required for microarray analysis. So I said, okay, this equipments are very expensive. Yes, really expensive, because you need minimum 2.5 crores to 3 crores, okay, to set up a complete the microarray working station. So this is the first one is hybridization oven to facilitate the temperature and the conditions for hybridization. The hybridization means joining of two single stranded molecules into one double strand molecule, and a fluid station to wash your chip. The biochip can be inserted here, and that will wash with the cold buffer. And the third one is the bigger budget, okay, that is the scanner and CPU with the software. Okay, all these technologies related to a particular company. Each and every company, they have their own software and the scanner type. And so next one, okay, this is the methodology, and you saw the animation type. Okay, this is the actual type, and the loading and the labeling of DNA, the control DNA, and this is hybridization oven, and we can keep the chip okay inside for hybridization. And after hybridization, just you can wash it in the washer, and after that you can insert after drying, the blowing the air, okay, just to dry, you should not heat it, and you can insert into the scanner. And finally the data analysis, the data can be retrieved from the software. And the applications are n number of applications when compared to this technology. No other technology is equivalent to examine okay the gene expression changes and the diseases and the development and progression of the disease condition. The progression means okay if you're identifying it as a cancer means and what is the stage of cancer, maybe whether they started the cancer or otherwise maybe in the middle stage or maybe in the final stage of cancer. And to examine the targets for drug discovery, you can identify the new targets. So that is our focus, my focus, to identify the targets for the disease and to identify inheritable markers. Okay, you can do this type of one. Maybe we have flu chip, and also okay, all the chromosomal abnormalities. We can have another chip also. So mostly, okay, I did maybe a lot of research, okay, in this area, okay, like microarray research in Libya, because they had okay a lot of instrumentation and part, and mainly they are using okay this one for inheritable markers. So there is a chromosomal abnormalities, because all the Western countries or these complications are much more, and before marriage, okay, they will check it. In India, okay, it is not possible. And to detect viruses and other pathogens, okay, from the blood samples. So this also, that is the clinical microbiology also. Clinical molecular microbiology. Okay, there is another field is growing now, and that is the one, okay, we are going to use it now, okay, for the COVID-19 diagnosis. And advantages is easier to use, high throughput. You can analyze thousands of genes or markers at the same time. If you want to test only the blood glucose, you can test only the blood glucose maybe for half an hour. But this one, within maybe the fraction of seconds, 3 seconds or 4 seconds, automatically you can scan everything. And before scanning, you have to keep it for incubation and the hybridization oven for 7 hours. That's all. So maximum within 10 hours, you can get nearly the 5 lakh different test reports in your hand, and generate large quantities of data in lesser time, and do not require large scale sequencing. No need of sequencing at all. Okay, just we need mRNA and convert mRNA into cDNA. That's all. And allow the quantitation of 1,000 of genes from many samples. And okay, that's all about okay the DNA microarray. And we are coming to the focus of today's webinar. Peptide microarray. Just I said, okay, the peptides are used for diagnosis, and here we are going to use the peptide microarray for COVID-19 diagnosis. So now this is, and we have okay the very difficult time now. For the past 3 months, okay, we are suffering a lot, and now this is the peak time in India, especially in Tamil Nadu and all the places of Tamil Nadu also. Now it is spreading like anything, okay, due to the ignorance of the people. And the central government and down to WHO or state government, okay, they gave very clear instruction to all the citizens of India, and it is more contagious, and it will spread only by okay the respiration, or otherwise a person is very close by. But we are not following the steps, and still we are going to the market, and we are okay very near to the persons, and we don't know whether they have the symptoms or not. So that is a main problem. So know what to do. So how to diagnose okay all the persons? Is it possible to diagnose all the persons of a particular city like Krishnagiri or Coimbatore or Madurai? Is it possible? It's very expensive, and it will take okay a lot of the manpower, and also the cost is more, and also it need more time, laborious process, because we need the qPCR, that is the RT-PCR technology. And this peptide microarray is an alternative to RT-PCR technique for the diagnosis of COVID-19, and the higher sensitivity than RT-PCR. So that is the main point, and very less possibility for false positive and false negative results. For example, if they are giving the positive, and maybe whether it is sure, okay, whether the positive is positive, and if they are giving the negative, whether what is the surety is that the negative is negative. So that is why there are two possibilities, false positive and false negative, and we can avoid okay this false positive and false negative in peptide microarray, and this is cost effective for the test. That is, for example, if you want to pay for 2,500 rupees to 3,500 rupees for qPCR, and you can pay only 1,000 rupees for this. But the problem is, okay, the machinery we need in crores, and only one-time investment. And if we have the microarray workstation and all the scanners and all the tests are very cheap, nearly 60% cheaper when compared to qPCR. And the possibility of detection of further subtypes in one check. Now, okay, we have the qPCR test kit, and maybe okay, we have a particular strain in Coimbatore. Maybe now we can, you already know that okay, in China, another type of okay, the stream of infection is happening. We don't know okay that is the same strain or not. Okay, if corona is having COVID-19 is having okay more than 20 to 25 different strains, we can identify all the strains in one test. So that is possible only in peptide microarray. This is not possible in RT-PCR. RT-PCR means you have to do it 32 times. If you want to do it, 32 strains means 32 times you have to do it. Just imagine 32 times into 2,500 rupees or 3,000 rupees, how much? It is a huge amount. So that is why we are going for this. There is a company, the patented company, the Pepperprint. They're all very good people, okay, in Germany. Okay, they are all collaborating with us, and so they are doing a great job, okay, in this contribution to the diagnosis, molecular diagnosis of COVID-19, especially using peptide microarray. Just to have a look, okay, this video. Yeah. So this company, this paper chip, and developed by mm, this company in Germany, and they developed the kit, okay, for the COVID-19 diagnosis. And this one is maybe possible for all the different types of diagnosis, including cancer diagnosis and maybe molecular aberrations or otherwise the chromosomal abnormalities, any type of diagnosis, okay, we can do it. So they developed a kit, okay, this kit having a small microarray slide. So each microarray slide on the back side, okay, the microarray ID can be given. So this is exclusive for only one patient. And so this one is approximately having so many number of epitope peptides. So just you can have it, okay, here the sequences of ORF1AB polyprotein of COVID-19. You know very well, okay, this is a RNA virus, and this virus, okay, having so many proteins like ORF protein and surface glycoprotein and envelope protein, membrane glycoprotein, and ORF6 protein, and so many different types of proteins are available. Okay, and this elongated protein sequences were translated to 4,883 different 15 amino acid peptides and printed in duplicate, okay, in all the microarray plates. So that means each and every one we are going to follow the statistical analysis in duplicate. Okay, and here I'm going to show that the database of the COVID-19 genome sequence, and this is a hyperlink. I'll show a small tab. Okay, you can see here, okay, this is GenBank data, and this is the publication date is 18th March 2020. So this is the Wuhan sample 1, that is severe acute respiratory syndrome coronavirus 2, that is COVID-19, okay, the Wuhan sample. The first report of the gene sequence, this is starting of the gene to end of the gene. So this is a complete sequence, and based on that, the scientists developed the peptides, the complementary. By using the bioinformatics, we can develop the complementary, the DNA to the protein. Okay, this making of protein fragment, okay, from the DNA sequence. So this file, okay, will open, and this Excel file, and just you can have a look. This is a file of pepper chip and the peptide map microarray chip. So this is the one. I think you're able to see, okay, this one. So here in a particular chip, approximately 4,883, that is 4,883 total number of peptides are available. Okay, and overlap is 13 amino acid, and the peptide length is 15 amino acid. It is a peptide. Peptide means only 15 amino acids are there. The total 108 spots for each and every spot, and this is the peptide. Okay, and you can see how many spots are there. Okay, and this is a peptide list. So this is ORF NAB protein and polyprotein sequences. And similarly, okay, this spike protein will be there, and you know these many number of the proteins are present. The nucleic acid, phosphoprotein, and all the proteins are covered, okay, in this. So there is no missing of data, and we can get everything. So this peptide microarray epitope plot, maybe if you see the overview of the complete mapping of all the peptides, we can see, okay, this one. So this is a hyperlink for, yeah, this is a chip. Just imagine. So within the chip, small dots are there. And if you are zooming this one, I'm going into the chip. Can you able to see the different numbers? Okay, the G, S, G. So all these are peptides. Maybe you can see one by one. So total 4,883 is there. Okay, this is an overview of peptide microarray. Now we can understand the complexity of peptide microarray, and the preparing the microarray plate is a little difficult when compared to anything in the world. Okay, and we cannot prepare the chip and all. We can buy the chip from the companies because we need some robotic system to prepare the microarray chips and all. Okay, the main strategy behind the creation of chip is the total proteome, the sequence is retrieved from gene bank, and they will develop the peptide. That is, peptide is synthesized, okay, by peptide synthesizer, and they will create an overlapping peptides, and they will implant, okay, on the microarray plate. And after that, and this is a demo, okay, how they are going to do, okay, this peptide print, okay, in the experiment. For example, if you are getting a pepper print microarray chip, and if you are doing the COVID-19 diagnosis, this is the video. Just to have a look.
Peptide microarray analysis involves various lab processes. We're looking at
I think I hope okay everyone understood the concept of peptide microarray, and this is a article published already okay in the year 2020 with coronavirus identification by using SARS coronavirus identification by using the proteome, and this is the peptide like construction of a peptide library, overlapping peptide library, and this is quoted with all the. This is the principal concept, and the concept is adopted by the company. Okay, there is microplate reader, and this is the peptide microarray okay in COVID-19 diagnosis. So these are all the areas. Just you can see the yellow color is the peptide sequence that is 66 to 70 amino acid, and all these areas are the epitopes. Epitopes it means okay the antibody binding sites are called as epitopes. So they selected okay only these for a particular protein, that is a nucleocapsid phosphoprotein of COVID-19, and this is also COVID-19 spike protein. So this is the main problem because this is going to bind with okay, ACE2, okay, angiotensin conversion enzyme 2. So this enzyme is responsible to bind with our cells okay by the virus particle. So these are all the indications, and this one having so many number of epitopes. Just you can see 1 2 3 4 5 6 different types of epitopes are there, and for all these epitopes will give the significant the role okay in the diagnosis. For example, okay here okay, this is the overall chart of the human IgM antibody and IgG antibody. You know very well okay what is the difference between IgG and IgG. IgG is simple and IgM is a little complex, and both are produced okay in the patients, the COVID-19 patients. And you can see here the red color indicates these are all the presence of IgM antibody okay towards a particular the peptide sequence that is an epitope, and this one is a major one. So this P45, P45 means okay this protein 45 is very much involved, and the amount of antibody formation is also more okay based on the Z score. So based on this, we can identify without any hassles, without any okay the false positive, false negative, and so we can conclude whether this patient is affected by 100% corona positive or corona negative. So this is possible only with peptide microarray analysis, and unfortunately, we don't have this type of the technique okay in India. So very few maybe advanced hospitals okay they have this technique. Okay, they got the samples from the missionaries from Pepperprint, and they are using it. Unfortunately, in South India okay we don't have, and so this is the potential neutralizing antibody produced by our immune system. You can see here this is the very important epitope. Okay, this epitope is a target. Okay, maybe so these are the antibodies present in our serum, and this will go and bind to the epitope tagged okay with the microarray plate, and this will give the signal. So based on the signal okay we can identify, and this is for your information okay this is the spike protein, and this area the red color area is the epitope. So what we are doing is exactly right, and yeah, this is the final ultimate result. The SARS COVID-19 infected person's microarray plate working by pepper chip SARS COVID-19 okay the protein microarray scan. This is the healthy control. You can see the difference. So many antibodies are produced, and these antibodies binding to the sites, and this will give a signal, and the signaling pattern is different, and here this is different. So based on that, we can identify whether the patient is affected by COVID-19 really or not. Okay, and these are the images from the courtesy from the paper chip material data sheet of paper chip. And finally, okay, with this technology, we can identify the person whether he or she is infected with COVID-19 or not. Okay, and with this, I conclude my lecture, and I would like to thank the Pepperprint also, because they are the leaders or pioneers okay, in this field, and they are supporting us. And one of my friend is there okay in Pepperprint, and he is also collaborating with us. And due to that only, it is possible to understand the concept of okay, the peptide microarray. And I thank Pepperprint also from Germany, and also I would like to thank our management, the same as the College of Science and Commerce, for giving the opportunity to deliver this webinar to the Government Arts College, the PG and the Research Department of Biochemistry, and especially to my mentor and my guide and all. Okay, and she is the one, okay, supported us, and I know very well. Okay, Jayanti madam is very keen on clinical biochemistry. Still, I remember her classes also. So thank you, Jayanti madam, okay, for giving the opportunity, and thank you, principal of Dr. Jayanti madam, the principal of Government Arts College, Krishnagiri, for giving the opportunity, and all the organizers of government college. So they're all okay supporting okay for this, and only because of them okay this is possible. I thank everyone, and not but then the last but not the least, the students. Okay, we are all here because during the corona period, and you are in the home also, where you don't know okay this bandwidth to see all the slides, but you're all very keen attending the webinar. Okay, and I thank you also. Okay, all the best. Thank you, madam. Thank you, Anand.
Thank you, sir, for your
Thank you, madam.
Now, Dr. Srinivasan, sir. Hello.
Hello.
Dear participants, you are all requested to come forward with the questions. Sir is welcoming to answer you.
Hello, doctor.
Yes, sir.
Hi, I'm Dr. Murthy.
Yes, sir.
A few questions from my side. As
Yeah,
you
please.
mentioned during the presentation,
Yeah.
by using the biochips, analyze the more parameters in a very short span of time. Okay, as you mentioned, more than 1 lakh or 2 lakh parameters we can analyze.
Yeah.
Okay. Can you discover some major laboratories following this type of diagnostic tool, using this tool in India?
In India, we have very limited, sir. That's why I said, okay, because we need, okay, some sophisticated instrumentation facility, because we have to spend, okay, 2.5 crores to 4 crores. And I know the research is going on well. And for the cancer chip is ready, and the flu chip is also ready. Maybe if you're going to US or Canada or USA, all the developed countries, okay, they made it already, and they are using at present.
So
Okay,
I am
but unfortunately, in India, we don't have okay this support okay from the government, and we need okay more instrumentation okay for this.
Yeah, absolutely correct, because it is minimize the TAT time, no turnaround time.
Yes,
When
yes, and you know the
numbers
main problem is if you're using this one, you can pay only 1,500 rupees to 2,000 rupees for all the 2 lakh 10,000 or 5 lakh test okay each and every time. And no need to pay okay each and every time okay maybe for glucose test or otherwise creatinine test for each and every 150 rupees or 200 rupees. So this will add your budget. Okay, and this is a wonderful technology, and if it is comes to okay all the parts of India, comes to all the each and every segment of hospitals, especially in the developed hospitals and also in government hospitals, so this may be very useful to the community.
So any other country is following this method of
all the developed countries, including because I was there in Libya, so they have the flu chip. Because a person is coming to Libya means okay, they may be advised to go to the a small clinic okay there in the airport, and they have to give the sample, and we have to wait for okay maybe 1 hour, 2 maybe 1.5 hours or 2 hours okay to get the results, and based on that we can we go to the particular university or to the place in Libya. It was before, maybe we were there okay in Libya for 4 years. At the time okay the SARS virus was there. At the time okay we faced okay this problem. So they used this technology, I'm telling okay 2007 to 8. So now the duration is okay now the 2020. Just imagine okay still we are in the backlog.
Okay, thank you for your
Okay, sir. Next question. Any questions from the audience?
Hello, sir.
Hello, madam.
Hello.
Can you share the Excel sheet which you showed during the presentation?
So you want to see the Excel sheet?
Yeah.
One second. Can you able to see my window?
Yes, sir. Sure.
Yes. So this is the Excel sheet you asked.
Can it be shared, sir?
So you want to see this one? You
No, I'm
want to
asking if
get
it
this
can
one?
be shared.
Yeah, sure. Okay, I'll share it in the chat box, I think, if possible. Or otherwise, I will give it to Jayanti ma'am, and she will give it to you.
Sure, sir. Thank you.
Okay, and maybe the last slide I gave my Gmail ID, and you can request, and I'll send this one to you.
Okay, sir. Sure.
Or
Thank you.
otherwise, you can go to Pepperprint. Everything's available. Okay, all the videos and the YouTube, and they are helping to the society. Okay, everything is available. Okay, I'm not having only the secret document of peptide microarray of COVID-19 diagnosis. So this is open to all. Okay.
Okay,
And
sir.
please visit the Pepperprint website and go to the different segments. There is separate segment is there, COVID-19, and you can download all the data sheet and the worksheet also, and also this document is available.
Okay, sir.
Okay. And here, okay, there are two sheets of there. Peptide map is there and peptide list is also there. This is list and this is map. You can get all the informations.
Right.
Is it okay?
Yeah, thanks a lot, sir. That helps.
Thank you. Thank you. Thank you for your question. Yes, any more questions?
Any more questions, participants? Please do interact.
Maybe in the chat box, whether they are typing it.
You can present the message in queries in the chat box also.
Sir, may I know the application of glycomics? Yeah,
Application
good
of
question. Glycomics is a separate area, study of carbohydrates and its role in the normal metabolism and also, okay, in the normal physiology of cells. Glycomics is the data and the study of data and interpretation of data of the glycan chains, and that is having, okay, a lot of opportunities okay, in the near future. And those who have the experience in glycomics, you have a lot of scope in abroad. In India, very few scientists, okay, they are doing the research, especially in the Indian Institute of Science. Okay, one famous professor is there, and also in South Campus, Delhi. Okay, one professor is there. So they are doing a lot of research work in glycomics related to microorganisms. So not with animal cells, and so very good scope. Okay, for glycomics. Any more question? What about the specificity of transcriptome analysis? The transcriptome analysis is entirely different when compared to peptide microarray. The transcriptome, okay, you need maybe more sophistication because you are going to deal with mRNA. Okay, and here we are going to use only the proteins. So this all these proteins not spliced from the proteins. Okay, the peptides are not spliced from the proteins because the splicing and also the purification of the peptide fragments, it's very difficult. You know very well the protein purification is one of the hardest area in biochemistry and also the bioprocess technology. It is very difficult. Okay, the bioprocess technology, that is downstream processing. So instead of that, okay, mostly all the companies, they are able to produce or otherwise synthesize the peptides. Okay, so that is why they choose, okay, the synthesis of peptides, and they will tag it on the microarray plates. The transcriptome analysis, that is another version, and you need a lot of skills, okay, to do that.
Any more queries?
Yeah. If you have any questions, please type it in the chat box.
Any more queries? Is there any queries in chat box, sir?
Till now, all the queries I answered, madam.
Answer. Thank you so much, sir.
Thank you,
Now,
madam.
call upon Dr. Srinivasan, sir, Associate Professor, Department of Biochemistry, Government Arts College for Women. He is my colleague. He is here in the platform to present the vote of thanks.
Thank you, madam.
Thank you, sir.
Good afternoon all. On behalf of Department of Biochemistry, Government Arts College for Women Krishnagiri, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed this webinar on peptide microarray and COVID-19 diagnosis organized by our institute. At the outset, I thank our chief guest and resource person, Dr. T. Vinod Kumar sir, Associate Professor and Director, School of Biological Sciences, CMS College of Science and Commerce, Coimbatore. We are really enlightened with our knowledge and presence, sir. We are thankful to our Honorable Principal, Dr. R.R. Jayanti Madam, for her motivation. I would like to thank our HOD, Dr. G. Jayanti Madam, for his enthusiastic support. A special thanks to the organizing committee, teaching staff for their unflinching support and coordination. Our heartfelt thanks to our students for active participation. With their warm words and kind message, we move to end up today's webinar. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, ma'am. Thank you, sir.
Hello, sir.
Thank you for your support.
On behalf of the department, I also thank you once again for giving this presentation in the right time. Very kind of you.
Thank you, madam. Actually, this one can be extended to one more half an hour because I restricted the time because today is Saturday and the students can be maybe they can attend the webinar maybe for 45 minutes. That's why I limited my presentation. Thank you. Thank you, madam.
Thank you,
So
sir.
very nice to interact with you all the faculty members of Government Arts College and including the principal ma'am. Thank you. Thank you.
I thank one and all once again the participants of faculty, participants and others for the seminar and highlighting this webinar series. Thank you one and all.
Thank you.
Thank you, sir.